Hello everyone, my name is Rahul. In the previous video, we had discussed about plastids. In this video, we are going to discuss about ribosomes. Ribosomes are non-membrane bound granular organelles that are seen in both prokaryotes and eukaryotes. These are non-membrane bound. This is why they are also seen in prokaryotes, where all other membrane bound cell organelles are absent. Ribosomes, they were first discovered by George Palade in the year 1953. This is why ribosomes are also known as Palade particles. Ribosomes are also known as protein factory of the cell because the function of protein synthesis is carried out by ribosomes. Ribosomes are mainly composed of ribonucleoproteins, which means they are made up of RNA and proteins. Now there are two types of ribosomes. One is the eukaryotic type and other one is the prokaryotic type. The eukaryotic ribosomes, they are 80S ribosomes, and the prokaryotic ribosomes, they are 70S ribosomes. Now these 80S ribosomes, they are seen in cytoplasm of eukaryotic cells. The 70S ribosomes, they are seen in cytoplasm of prokaryotic cells. As well as mitochondria and plastids of eukaryotes. Now here in both 80S and 70S, S stands for Swedberg unit, which is an indirect measure of density and size. Swedberg unit is an indirect measure of density and size. Now what is the role of magnesium ions? Magnesium ions are responsible for joining the subunits of ribosomes. Now ribosomes, they contain two subunits, one larger subunit and a smaller subunit. Magnesium ions, they are responsible for binding the ribosomal subunits. These ions, they form a bond with the phosphate groups of ribosomal RNA of the two subunits and thus help in keeping them together. A minimum of 0.001 mol Mg2 plus ions are required in a cell to keep the ribosomal subunits bound together. Now talking about the chemical composition of ribosomes, the 70S ribosomes, they are made up of two subunits, that is 40S plus 30S, and the 70S ribosome, it is 60% ribosomal RNA plus 40% proteins. The 80S ribosome, it has two subunits, namely 60S and 40S, and it is made up of 40% ribosomal RNA plus 60% proteins. The 60S subunit, it is made up of ribosomal RNA, that is 28S, 5.8S and 5S. The 40S subunit is made up of ribosomal RNA 18S. The 50S subunit, it is made up of ribosomal RNA 23S and 5S. The 30S subunit, it is made up of ribosomal RNA 16S. Now talking about the functions of the subunits, the 40S subunit, it is responsible for formation of pre-initiation complex which is required for starting the process of translation. So for the process of protein synthesis to begin, a pre-initiation complex is required, which is formed by the 40S subunit of the ribosome. The 60S subunit, it is involved in, firstly, along with pre-initiation complex, it forms the 80S ribosome, that is the complete 80S ribosome assembly. It is only possible after formation of pre-initiation complex and its joining with the 60S subunit. That is, along with pre-initiation complex, it forms the 80S ribosome. That is, the formation of complete 80S assembly is possible only when the pre-initiation complex binds with the 60S subunit. It also provides A, P, and E sites during translation. The third function is it also helps in peptide bond formation during translation, and thus it helps in protein synthesis. Now there is an entity known as polyribosome or a polysome. So at the time of protein synthesis in eukaryotes, multiple ribosomes they get attached to a single mRNA molecule. Such an assembly is known as polyribosome. This polyribosome, it is not seen in prokaryotes. This is not seen in prokaryotes. With this, we finish off with our discussion on ribosomes. See you in next video.